is Chandrima and I am doing my masters from Indian Institute of Technology Varanasi, and today we will be talking about one of the most headache giving topics in JEE Advanced, which is biomolecules. And from the word biomolecules we can see that it's bio and molecules. So basically biomolecules are those molecules which are present in the biological systems. Now, for example, we can say lipids, carbohydrates, proteins, nucleic acid, and vitamins. What are lipids? Lipids are basically the fat in your body. They are known as the lipids. What are carbohydrates? They are the sugars. Okay. What are proteins? They are the building blocks of our biological system. What is the nucleic acid? It is like the genetic information, carries the genetic information. And what is vitamins? It's the micronutrients which are present inside our body. But today in this lecture, we will be specifically concentrating on nucleic acid. So now we will talk about nucleic acid. So what is a nucleic acid? Nucleic acid are found inside the nucleus of the cell. What are its function? Its function is to transfer the genetic material. Now I'll show you how it is, and this is just for higher living organisms. There are lower living organisms like bacteria and all, so they don't have this, but still I'll explain much more later on. So this is a cell, and this, as I have labeled it, is the nucleus. And if we expand the nucleus, we can see that this is the nucleolus while this is the chromatin. The chromatin are thread-like structures, okay, but when we take a little portion from here or from here and expand it, we can see that this is a chromosome, and inside this chromosome again we can see many coiled structures, right? These coiled structures, when zoomed again, can be seen that they have DNA. Now, this is not the ultimate figure of DNA. We will discuss more about this later on. In the next portion, we will be discussing on the types of nucleic acid. So, in the last part, we discussed about the sugar, the pentose sugar. Now, we will be talking about the bases. Basically, there are two types of bases. One is purine and the other one is pyrimidine. And under purine comes two types of bases. One is adenine, which we represent by A. Second one is guanine, which we represent by G. Inside pyrimidine, there are three bases. One is cytosine, which is represented by C. One is uracil, which is represented by U. And one is thymine, which is represented by T. Now, there is a difference in bases in DNA and RNA. In DNA, we have A, T, G, C. But in RNA, we have A, U, G, C. Okay. Now we will see how to differentiate these bases, and it is very important to know the structure of these bases. So for the purine, just to remember how to remember the purine and the pyrimidine groups, like which one lies inside. Okay. So the name purine is small, right, compared to pyrimidine, but the structure of purine is bigger. So remember it like that. For purine, the structure is bigger, but the name is small. And for pyrimidine, the name is big, but the structure is small. Now, for differentiation between these two groups, okay, what do we need to do? First, in adenine, you can see there is a NH2, while in guanine, we can see that there is a oxygen. Okay. Next, here we can see that there is a hydrogen, whereas in guanine there is an NH2. Okay. So these are the only two differences in adenine and guanine. Okay. Now for pyrimidine. So as you can see here that for cytosine and the difference is first oxygen and NH2. Okay, and between thymine and cytosine and uracil is that there is a CH3 group. Okay, now we know that RNA has AUGC, means there is no T in the RNA, which means there is no thymine. Now we also call thymine as 5-methyl-uracil. Why? Because if we take the structure of uracil and put a methyl here, you will see that it is the same structure, or it becomes a thymine. Now this is just for general knowledge, but since the oxygen here is naked, right, there is no guard and any group can easily attack and remove this hydrogen. So RNA is more unstable than DNA. Now one of the more primary differences between purine and pyrimidine is that purine has 2 rings, okay, for both the bases, and pyrimidine has only 1 ring. Now the rings of the purine, this bigger ring for both adenine and guanine is known as pyrimidine rings, while the smaller ring is known as imidazole rings. Okay, and apart from that, in the pyrimidine, we can see that there is CUT, right? So for pyrimidine, you should remember that UT has 2 carbon and oxygen bonds like this. Okay, but for C there is one NH and one carbon and oxygen bond. These are the main differences between C and UT. And also, purine always bonds with pyrimidine. Okay, and it's not like adenine can bond with guanine. No, adenine has to bond with either uracil or thymine, and guanine always bonds with cytosine. There is no other way. Okay, now if the adenine will bond with uracil or thymine will depend on the type of nucleic acid. If it is a DNA, only thymine will remain inside the nucleus of the cell. There will be no traces of uracil, and in that condition the adenine will bond with thymine. Okay, and if it's an RNA, then the cell will have uracil instead of thymine. Then the adenine will bond with uracil. Now in the next part we will be talking about nucleotides and nucleosides. Now we will be talking about nucleotides and nucleosides. So what is a nucleoside? A nucleoside is basically sugar and base. Okay, so this is the methyl sugar and here is the base. Now depending on its nature or the type, that is if it's DNA or RNA, the OH or H will remain, and apart from that, in the place of the bases, either A, T, G, C, either of them will come here, or A, U, G, C, either of them will come here. Okay, and then there is nucleotide. Okay, so what does a nucleotide comprise of? Phosphate, sugar and the base. So this is the structure of a nucleotide. Now again, according to DNA or RNA, the bases will be there, that is either A, T, G or C, or A, U, G, C, and apart from that H or OH, and apart from that this is the phosphate group. Okay. Now how do we know the difference between, or remember the difference between nucleotide and nucleoside? Now as you can see, the nucleotide and nucleoside both has this in common, okay? Sugar and base. This is common. Only thing that is different is this phosphate. So just remember, if there is a S, it's only sugar and base. Okay? And if there is nucleotide, just add a phosphate. And if it's not a S, just add a phosphate to it. Okay? And apart from that, the nucleoside as you can see here, as I told you, since that is a common, then what we can say from this, that a nucleotide is basically phosphate and a nucleoside, right? Apart from that, so nucleic acids are basically polynucleotides. Why poly? Because it acts like a polymer, okay? And there are many nucleotides they join to form nucleic acid. Okay, so this is a representation of the nucleic acid, and this box here is one nucleotide. Okay, and this, like, since we can see there is sugar, phosphate, and this goes on. Okay, this part is known as a strand. On this strand lies the bases, that is ATGC or AUGC. Next, we will talk about the phosphodiester bond. So, when two nucleotides form a bond, they form this molecule. Okay. So now how do they form a bond? So basically, the OH here forms a bond with the oxygen here. Okay. And when they form a bond, this phosphate group comes here and joins these two molecules. Okay. And this is called the phosphodiester linkage. Okay. Now, why is it called the phosphodiester? First, ester. We know the structure of the ester group is C double bond O, bond C, right? And in here, this part, we can see that there is a phosphate group, and around it there is this one ester group and this two ester group. Thus, di comes, and since there is a phosphate group, that's why phosphodiester linkage. Okay. Now, next we will now talk about in details about DNA. So, this is the last part of the nucleic acid. Now, we already talked about DNA and RNA. Now we will see its structure. DNA has a double helix structure, okay? And as we have read previously also, there is a strand of DNA which consists of sugar and the phosphate, right? So, in this diagram, this part, okay, and also this part are the strands of the DNA which consist of sugar and phosphate, and all the strands are the bases A, T, G and C. Now we know that there is double bond between A and T and triple bond between G and C. Now what are these bonds that we are talking about? It's the hydrogen bond. Okay, so from here, as you can see, I have mixed all of the bases. Now we will do one very simple exercise. Okay, there is two bonds, so it can either be A or T. We will just write A here. Okay, here also two bonds. Let's write T. Here also two bonds. Let's write A. Here again two bonds. Let's again write A, and again T, and again A, again T, and again A. Rest here is a 3 bond. 3 bond is in G and C. So we can write G. Okay. Here we can write C. Here we can write G. And do we have one? No, we do not. So now pause the video and check it for yourself. But before you do that, remember A always comes with T and G always comes with C. So what will be here on the other side? I'll do one for you. For A will T, for G will be C. Now fill the rest here. I hope you guys have paused and this is the answer. Please check if you have done it correctly. Okay. Now, for the RNA is a single stranded structure like this one here. Okay, and in RNA again, so I have already told you before that in RNA there is no thymine, there is uracil. So between adenine and uracil there is a double bond, and between guanine and cytosine there is a triple bond. Now, since there is no complementary pair, we don't need to remember this much, but just remember adenine and uracil double, guanine and cytosine triple. Okay, double bond. So we will write a double bond. Let's say U. Okay, triple bond G. Again U. Again C. Let's say A. And this one is also a double bond A, or this one let's give it G. Okay. So you now know how to know where is what. Okay. Now RNA is of three types. The messenger RNA, which is also known as mRNA. The ribosomal RNA, which is known as rRNA. And the transfer RNA, which is known as tRNA. Okay. And in the last part of this video, we will like to conclude with the biological functions of the nucleic acid as a whole, not only DNA, not only RNA, but as a whole. So, what do DNA, RNA, or nucleic acids do? It's the chemical basis of heredity. Okay. Next, it preserves the genetic info. Okay. And last but not the least, it helps in protein synthesis, and protein is a very important natural building block of our body, right? So it is very important. And apart from this, if you still don't understand all this, just know that every character you got from your parents, it comes with the help of DNA, or yes, in your case it's DNA. For unicellular organisms, it's RNA. Thank you. I hope you loved the video. Please comment, like and share. Bye bye.